Yeah, because the main families are one is people are know about one is neurodegenerative disease. So they offered the A and whatever. I said, well, just eat for first they said if I wanted to, sure. I said, well, then I would contact the president of the society. And that's the time. I didn't know. Right, he was turning over to somebody else, so there'd be
no, I learned all this really very quick, and I thought that we were. In Rosario before the Rosario.
And then I'll go to
¿Y después de Montevideo?
Are you from Rosario?
Yeah.
You know, I have an Argentine postdoc he emigrated to Israel, but now he's originally from Chile.
I'm from Chile. In the south.
Yeah.
What's his name? Yes, well, intestinal.
Yeah.
I know him.
It's a small world.
Because I just when I was doing intestinal, I met him a few years ago.
That's right. Yeah, he was doing his PhD. He's really good.
Okay, so it's a pleasure to introduce Adrian Kreiner, who is going to talk today about the antisense phase modulation of SM2 splicing. So he's going to talk about some of your work in neurodegenerative disease. Adrian is originally from Uruguay. He went to the US to do his PhD at Harvard, no? Yeah. He was very young. Then he stayed in the US and 15 years ago, something like this, established your own lab, no?
25.
25, sorry. And he has done seminal contributions to the field of RNA splicing, and one of them is the discovery of ester proteins, which are key constitutive of the therapeutic splicing and constitutive splicing machinery. And today he's going, as I said before, to talk SM2 splicing and neurodegenerative disease, and it's really an honor, pleasure to have you here.
Okay, thank you. I'm very happy to be here. I work at a research institute, so we're not known for teaching a lot. We give lectures for seminars and so on, but we're not required to teach. But it's something I enjoy doing every once in a while. So I may be a little bit jet lagged, and so if my mind wanders and something is unclear, I prefer if you raise your hand or speak up and ask to clarify something. Don't worry about doing that. All right, so I work on RNA splicing mainly, but we have studied a lot of the basic mechanisms, and then at some point we were interested in the contribution of defective splicing to some genetic diseases and cancer. So we study that aspect in part in the lab to try to understand how important is splicing in the context of human disease. And then at some point we realized that in specific instances we could use that knowledge to try to fix errors in splicing in a targeted way for a particular disease. And I'll mainly talk about one disease, which is spinal muscular atrophy. But so I'm not exactly a neuroscientist. That should be very clear very quickly. But It'll be obvious why we're choosing to target that disease first. And I will give a little bit of background about the disease and about splicing, at least minimally. But as I said, just ask any questions at any time. So just to remind you that we're studying primarily splicing, which occurs in the nucleus, and that's required to remove all of these introns. And some of it happens in coordinated manner with transcription, not exactly in a linear sequence like is shown here, but if the gene has introns, then this is a crucial process, and it has to happen very accurately for correct gene expression. And what's shown here is an average gene. An average human gene has, I think, 8 exons, and but there's a gene with 363 introns or exons, and there are some that have no introns. But so this is sort of a representative human gene. And each intron is removed by this pathway, which requires recognition of 3 blocks of sequence motifs, one at the 5' splice site, one at the 3' splice site, and one at the so-called branch site, where this intermediate forms with the covalent bond there. So whenever we talk about splicing, it happens in these 2 steps, and you generate these intermediate that are difficult to find in the cell because they're not long-lived. And then this final product here is also rapidly debranched and degraded, and this is the product of interest. And so the splicing machinery has to recognize these signals and then do these 2 chemical reactions that involve these 3 regions of the intron. So the splicing machinery is very complicated. 5 small nuclear RNAs, something like 170 proteins in humans, plus some regulatory components as well. So a few of the components are shown in this cartoon. For instance, this is U1 snRNP, small nuclear ribonucleic protein. Here's a recent structure of that. The RNA part has sequence complementarity to the consensus 5' splice site. So it's one of the factors that recognizes the 5' end of the intron. Another snRNP, U2, uses also RNA base pairing to recognize the branch site. And these are the consensus motifs. I'll come back to that. And a heterodimeric protein, U2F65 and 35, recognizes the 3' splice site element. But then many of these other components, again, recognize these at different stages, and some of them allow remodeling of the entire RNA protein complex, and ultimately the catalysis that involves the 3 sites. As you can see from the motifs, the elements are degenerate in humans. So GU at the 5' splice site is highly conserved, but then at the 3rd position it tends to be a purine, and then it gets weaker and weaker. And there's a little bit of conservation at the end of the exon as well. The branch site is a very weak signal. The A is almost always an A, but not always. The 3' splice site has these pyrimidine tract, and then the AG is highly conserved. So these elements are very important for splicing, but they're not sufficient for the splicing machinery to know where the exon-intron borders are located. We also have alternative splicing. I know that you learned about it already, so just refresh your memory that many genes can generate 2 or more products by alternative splicing, and so I just mentioned general splicing, but in alternative splicing, you can either, for example, if you have this exon, you can include it in the message as indicated here, or you can skip it, giving rise to different isoforms. And these are the different basic patterns of alternative splicing. For instance, you can have 2 or more exons that function in a mutually exclusive manner. You incorporate one, but not the other, or vice versa. And these other methods allow parts of an exon to sometimes be omitted and sometimes be included in the final message. Again, the first exon and the last exon are defined if there's more than one promoter or more than one 3' processing site. And then long genes with lots of exons can use combinations of these patterns, and then you can get one gene generating many isoforms. So this is a very important source of the diversity of proteins encoded in the genome. informatics and RNA-seq analysis indicates that a lot of the human genes, most of them can encode more than one product, at least 2 isoforms. So quite a few of these have one abundant isoform, and the other is more difficult to identify in certain tissues or certain types of development. But it can be much more than 2 as well. And then these isoforms can perform similar functions. They differ in some subtle but important ways, or they have very distinct functions. If the splicing can determine where translation initiation occurs in the reading frame for translation, so you can have isoforms that have completely different sequence, even though they're encoded by the same gene. So there are extremes of that to completely unrelated products made by a single gene. Viruses, for example, use that a lot. The isoforms can antagonize each other's functions, so you can have a gene generate one important product, and the others may be just a side reaction or that decreases the output of the gene, or it could be something that has the opposite function as the main isoform. For instance, it may bind a ligand but fail to catalyze a reaction because it lacks a portion of the active site. Then it behaves as a dominant negative gene product. For instance, in apoptosis, you have isoforms that are pro-apoptotic and others that are anti-apoptotic. It's fairly common. Also, the different isoforms from the same gene may end up in different locations, both as mRNAs or as proteins within the cell or outside the cell. So this process gives a lot of versatility to gene expression. And typically, the isoforms that are made can be regulated, so different cell types or during development, or in response to signaling, you change the output of each gene. So for alternative splicing to happen, but even for constitutive splicing, the splicing machinery has to decide along an RNA, where are the exons? Is this an exon, or is it part of a longer intron? And after that, where are the exact boundaries? So that's determined by the splice sites that I already told you about. They can be strong or weak. That's a function of how similar they are to the consensus or how different. These other minuses and pluses signs denote regulatory elements that we call splicing enhancers, splicing silencers. They can be exonic or intronic. And so enhancers are recognized by RNA binding proteins known as splicing activators. Silencers are bound by repressors. And the overall structure of the RNA can modulate the accessibility of the splice sites and these regulatory elements to the factors, to the accompanying factors. So this is just a cartoon, but it's more or less what the field uses, the logic of exon identity, exon recognition. You have, let's say, here's a pyrimidine tract with somewhat weak elements, some purines instead of pyrimidines, so the interaction between this component is weaker than it might otherwise be. So this is not efficiently recognized as an exon, except that if an activator binds to an exonic splicing enhancer, it can help recruit the basal machinery. So these are just examples of such interactions. Protein-protein interactions can result in cooperative binding, or they can be indirect through coactivators that then interact with the basal component. Whereas repressors somehow repel the spliceosome from this region, and they can do that by antagonizing the binding of an activator, so these may be in competition for binding. The repressor is bound to a silencer element. But these activators and repressors, maybe different ones, can also bind to intronic elements and again affect everything. So this is a very simplistic model, but the idea is that there may be dozens or more of repressors and activators, a whole slew of such elements, and then everything is combinatorial. And then somehow the splicing machinery integrates all the information, and it's deciding in a case like this if this is an exon or it's just a long intron. So it's a binary decision at the end of that. So what can splicing mutations cause in this process? So even if you have a simple RNA, for example, the beta chain of hemoglobin, so this gene has just 3 exons and it's constitutively spliced. But the question is whether this is recognized as an exon or not. It's correctly spliced, maybe less efficiently so. But you have, this is fine, and now you retain an intron. This may not come out of the nucleus, so it won't be translated. If it is translated, it's going to lead to a truncated protein there or some other kind of problem. So that's intron retention. Another thing that can happen is exon skipping, because this 5' splice site is likely to be compatible with that 3' splice site. All of these elements tend to conform to the consensus sequence, so in principle they are compatible. That's usually the most common outcome. You have a mutation in a splice site, or it could be in this splice site or this one, you tend to get exon skipping. Another thing that happens, which is quite interesting, is that this site is no longer used, or no longer used efficiently, but then there's another sequence somewhere, for instance here, that also fits the consensus. The splicing machinery doesn't see that in this context. Here, it starts using it, and so it gives the wrong product because this exon is now smaller. This causes its activation of a cryptic splice site. And this product may be, in the case of beta globin, it may be roughly at 30% the level of this product. But here, you don't detect it at all, so it's kind of an interesting fidelity problem. So many mutations that lead to genetic diseases, you observe that, but even more often you see exon skipping. So how often does that happen? This is a review from 2007, and it looks at mutations in the human gene mutation database. Even at that time, or I forget when we extracted this number, there were more than 5,000 single nucleotide substitutions that affected the 5' splice site or the 3' splice site and caused a genetic disease. So probably a lot more than 10% of mutations are of that type in any genetic disease for which multiple alleles are known. If you also consider that this is kind of an obvious splicing mutation, but any mutation in the exon or in the intron could affect one of these regulatory elements, enhancers, silencers. I'll show you an example of that. Those can also be, even if they occur in the coding region, in reality they may be splicing mutations if they're changing the normal splicing pattern. So this number, and for some genes, half of the known mutations are causing missplicing. So it's a very prevalent problem. This is not a neurological disorder, but I picked this example because I think it's really cool. It's from a group in Tel Aviv. Maybe you know these people. You know everybody. So I think it's fascinating. So this is a disease where there's a complete absence of fingerprints. And they called it the immigration delay disease because at least when you come to the U.S., you have to show your fingerprints if you're not a citizen. So these people can't visit the U.S., that's for sure. What happens here is that in this mutations in this gene, loss of function mutations would be lethal, but in this particular case, it isn't because the gene produces all these transcripts by alternative start sites and 3' processing sites and alternative splicing. And so there's a particular transcript that's only expressed in skin, and so it has its own promoter there defining the first exon, and the 5' splice site of that exon, which is different from those, has a change from G to T at the first position. That's a very important nucleotide for the chemistry of the reaction. So this is effectively dead. And so since they're unable to make this product that only in skin, the phenotype is very subtle but interesting. I think there's only 4 families in the whole world with this phenotype. Did you know about this? Did you see this paper? Yes, that's very cool. Anyway, so I'll tell you about a much more serious disorder, which is spinal muscular atrophy. And this disease, so this is a neuromuscular disease, so it's a leading genetic cause of infant mortality. There's no available cure or treatment. The only thing that can be provided, for example, is ventilatory support and palliative measures. And there is, I'll explain, there's severe and milder forms of SMA. For the severe form, which is called type 1 SMA, half of the children die before age 2. Nowadays, with very aggressive therapy, not therapy, but assisted ventilation and nutrition and so on, they can live longer, but And then some of the older children here have milder forms of SMA, but they're able to walk, for example, at first, but gradually lose muscle strength, and their muscles atrophy because of loss of motor neurons. So ultimately, that they have to use these electric-powered wheelchairs that they control with one hand. So, and you can see some of them have spinal deformities, and all due to very progressive muscle atrophy. So it's kind of a heartbreaking disease. And these are children with a somewhat less severe form called type 2 SMA, and so these are very expensive wheelchairs, but they're able to move around very, very rapidly and very precisely with these. But they have, for them, it's hard, for example, to lift the arm, but they do have enough dexterity to do that. So mainly it affects children, but there is also an adult onset form. The key feature of SMA is the primary manifestation is a progressive degeneration of lower motor neurons in the spinal cord, and it's fairly frequent. It's estimated to be 1 in 6 to 10,000 live births in all populations. It's autosomal recessive. You'll see that it's a bit complicated, but in effect, it's inherited like a simple Mendelian disorder. And there's a variable severity, which I'll explain, but type 1 is the most severe form. Type 4 is adult onset SMA. 2 and 3 are intermediate severity. All of them are due to loss of function mutations in the SMN1 gene that stands for survival of motor neuron. And the product is SMN. And I won't talk about, we don't study ourselves the function of the protein, but it's an active area in the field. This just summarizes. It's a chaperone that catalyzes the assembly of the snRNPs that are involved in splicing, but probably other RNA protein complexes as well. So it loads a set of proteins onto the snRNA, and since these function in splicing, it's not a splicing factor, but it could potentially indirectly contribute to splicing. As far as where it's localized, you see it in the cytoplasm, but you also see it in the nucleus in intense regions that are called gems, but they're either similar or indistinguishable from Cajal bodies, which used to be called coiled bodies. In neurons, they're present in the axons as well, and they've been described to be moving bidirectionally along axons. And under stress conditions, they accumulate in stress granules. The gene product is essential at the organismal level, at the single cell level, but even though patients can be alive with complete loss of this gene because there's another paralog called SMN2 that's very similar, but it's only partially functional, and therefore they're not really healthy if they only have this gene in a functional state.
Is it known why it affects the motor neurons specifically, or because it looks like it could harm any kind of cell?
Yeah, that's a key question in the field, and it's been debated, and so people talk about whether this is the critical reason for it. I mean, this is considered a housekeeping function, and so why? I mean, that's a very important question, but nobody has the answer, and I think our work makes it more confusing. So we'll definitely discuss that. But it's common in human genetics to have a very, let's say, directed defect in a particular cell type, even for a ubiquitously expressed gene. So it's an interesting problem. That's what I think.
If you do like RNAi in cell culture,
you
have a phenotype, different cell types.
Essentially, by any knockout method will kill the cells, whether it's a liver cell or anything. Even in POMB, which has something related to this, it's essential. But there are threshold effects, so we don't understand why motor neurons are more vulnerable. I think it's probably a very complicated process, and we'll come back to that. Is that a cell autonomous defect? People have speculated, well, motor neurons are very large and they're very metabolically active, and all of that is true, but I mean, some sensory neurons are very large too, and why don't they die? And maybe they do, but I think I'm beginning to think that it has to do more with the interplay between motor neurons and other parts of the nervous system and even outside, and for some reason I have to explain why we have two genes. So this is evolutionarily quite interesting. So if we look in chromosome 5, that's where these genes are located. This entire region is a repeat, an inverted duplication. So this is a paralog of that, but this is a paralog of that. And so some of these are pseudogenes, but there's this clear inverted structure. This happened in primates. So you see this in monkeys and humans, but you don't see in other mammals. You only have this part and this antennic region. So you can see that, for instance, this is clearly a pseudogene. I wouldn't call this a pseudogene, but it's sort of becoming a pseudogene, or evolutionarily speaking, and I'll explain that. But another important feature is that because of the inverted repeat structure, this is very unstable in the genome. So there's copy number variation. So if you look at the chromosome 5s from different individuals, or the different paternal and maternal copies, you can have gene conversion, for instance, so 2 copies of SMN2 instead of 1 of each, or vice versa. I mean, that would be in healthy individuals. So some people, possibly in this room, don't have SMN2, but it doesn't matter. I mean, this is a kind of a backup gene, as long as you have a functional SMN1 gene. You can have expansion, so there could be 4 copies of SMN2 on this chromosome. And then depending on what you have on the other chromosome, it is a question of gene dosage. And this is, as you'll see, a only partially active gene, but if you have more copies of it, it can make up for the absence of that more effectively, and therefore the disease severity would be reduced. This is the domain structure of the protein. There's a bunch of domains. Again, I'm not a specialist in this because we haven't studied ourselves the function of the protein. I'm more interested in the expression of this gene and how to fix it. Underneath here are point mutations that cause SMA. So these are various parts of the protein. They indicate regions of the protein that are important for its function. There are loss of function mutations to various extents, so some are more severe than others, but that's in turn modified by the copy number of SMN2. But much more common in these point mutations is the complete loss of SMN1 by this gene conversion, which is essentially non-allelic homologous recombination. It can take out the whole gene or even just parts of the gene. And anyway, this is the structure of full-length protein as long as all the exons are included. So we're going to talk about exon 7, but in reality, since there's 2A and 2B for historic reasons, exon 7 is the penultimate exon, but it's really the 8th exon. But the nomenclature is that this is known as exon 7. So if we look at the exon structure of both genes to compare them, I'm not showing exons 1 through 5 because there are no differences there. There are only 11 nucleotide substitutions between SMN1 and SMN2. They occur in this intron, and one of them is in the last non-coding exon. And then there's this C to T change at the 6th position of exon 7. Exon 7 is 54 nucleotide long. And when I first became interested in this field, when these studies came out, what they showed is that they make chimeric genes between SMN1 and SMN2, and they showed that of those 11 substitutions, this is the only one that really matters. Because what happens is that exon 7 is a constitutive exon in SMN1. All the RNA essentially includes exon 7. This is a natural stop codon, and you get full-length SMN protein. But when you look at SMN2, this is an alternative exon. Most of the mRNA, or at least 50% in some cases, in some cell types, it's more than that, skips exon 7. And therefore, when this is translated, the last few amino acids come from exon 8. And that means the protein has different C-terminus. And it's called delta 7 because it lacks this portion. The main problem with this protein is that it degrades very rapidly. The half-life is not shorter than that of the full-length protein. Now, if you learn the genetic code, PPC and TPP are the synonymous codons for phenylalanine. So some of the mRNA still splices by including exon 7. And then this makes exactly the same protein, full length. There's no other difference. but there's less. And so this essentially is like it's not there because it's trashed very quickly. If you don't have this gene or you have mutant versions of this gene, then this is making the right product but in reduced amounts. And we come to your question, why do only motor neurons apparently care? First of all, I'm beginning to doubt that that's correct. I mean, the most obvious feature of the disease is the progressive loss of motor neurons. But let's say that you could fix that. I strongly suspect that other systems would begin to fail. But for whatever reason, some cell types are less worried about how much protein you have. Some sort of threshold amount. As long as you're above that, you're okay. This variability is kind of a boring explanation, but it could also have something to do with it. Some cell types end up including more efficiently because of the factors that are present that control that. Therefore, it's above threshold, and maybe in motor neurons it's closer to 10%. And that's kind of a quantitative explanation. I'm not sure if it's right. Well, then I should say that I described the function of SMN that's considered the housekeeping function, assembling nerves. But what's it doing on the axons moving up and down? So people have speculated that it's involved in MRNP transport to dendrites, and it's possible, but there's no functional experimental evidence of that. If that were the case, then you can begin to imagine that there are neuron-specific functions, although I'm not sure why they should be motor neuron-specific functions. All right, so our goal has been to first to understand why exon 7 is skipped, and then to see if we can do something about it. All patients have some copies of SMN2. If we include exon 7 more efficiently, either in motor neurons or wherever it's required, they'll make more full-length SMN protein, and hopefully that can alleviate the phenotype or arrest the progressive nature of the disease, or maybe reverse some aspects of it. Essentially, the most severe patients have at least 2 copies of SMN2. Very mild cases of SMA may have 6 copies or 8 copies because of this copy number variation that I mentioned. The other thing I should mention is that in mice or other mammals, non-primates, there's only one gene equivalent to SMN1, and there it's called SMN. And even in monkeys, you have the duplication, but you don't have the C to T change. This is a very recent thing that's only seen in humans. So when we try to use monkeys, for example, as a model system, you can't really use it to look at phenotype, and you can't fix splicing because it's not defective to begin with. So that's maybe somewhat unique to this disease.
Right.
Yes.
What is the mutation in SMN1?
What's
the mutation?
Right. Yeah. So the most common is a complete loss. That this gene becomes that by recombination. Or parts of it are gone that span exon 7, so that's effectively the same as converting the whole gene. And there are point mutations also, but that's dependent. So those are rare alleles, so I think I have, all of these are point mutations in particular alleles, but each of them is not particularly common.
They don't cause disease?
Yeah. I mean, these are probably all loss of function mutations, and then the severity of the disease is more a function of how many copies of that are present. I'm not going very fast, so you can tell me when you want to pause, etc. But otherwise, okay. All right. So this I'll go through very quickly. We investigated why the mutation causes exon skipping. This is the beginning of the sequence, and we studied an activator. The name has changed, but now it's called SRSF1. It likes this motif. That's not a perfect match to it, but the C is quite important, and when it changes to a T, this no longer binds. And so we think that's contributing to the inefficient recognition of exon 7 in this context, SMN2. There's also evidence that this change simultaneously creates a better match to the motif for a repressor called HNRMPA1, and so possibly both things are happening to some extent. But the main important thing is that the exon is no longer recognized efficiently when you have the U. So our strategy for fixing that, we've tried a number of things. We've been working on this for about 6 years now. So the idea goes back to that diagram I showed you earlier, which is whether this is seen as an exon is the the synthesis of the effects of all these combinatorial molecules, repressors, activators. The cell cares about the overall, the net effect of that. And so we have lost an activator that no longer binds there. But if there's a repressor somewhere and we could prevent it from binding, and the way we try to do that is to bind an antisense oligonucleotide at that position. Now, even though the activator still fails to bind, now the repressor doesn't bind either, so overall, this may be recognized more efficiently and you get more full-length mRNA. The way you get an antisense oligonucleotide to bind is simply by its sequence. It recognizes its target by Watson-Crick base pairing. That's, if you have enough length, then that's one of the most specific interactions in nature. So this can be a very targeted approach. So we do antisense technology, and we collaborate with a company that specializes in antisense, ISIS Pharmaceuticals. Even was a postdoc in my lab who did this work. He's done amazing stuff with Sophie. And when you do antisense, you generally use modified, it's like RNA or DNA, but you introduce modifications for a variety of reasons, and there are many choices there. We use this chemistry here. If you're familiar with nucleotide chemistry, instead of an OH as an RNA or an H as in DNA, we have this ether group here, 2'O, 2-methoxyethyl, or MOE, or MO for short. Instead of an oxygen here, there's a sulfur group. This is called phosphorothioate. And instead of cytosines, we have 5-methyl cytosines. And this is a reflection of many years of work at ISIS, like 20 years of trying many different chemistries for many different reasons, not really splicing, but we capitalized on that. The sulfur groups, for example, facilitate the carrying of this in plasma by interaction with proteins. These modifications stabilize against nucleases. The methyl cytosines are introduced there to avoid inflammation processes. And also the presence of these groups facilitates the cellular uptake of the oligonucleotides through receptor-mediated endocytosis. And so when we deliver this, all we have to do is dissolve it in buffers, saline, and then that's it. In vivo, at least, it's fairly efficiently taken up by many tissues, many cells. Once it does that, the charge facilitates its migration to the nucleus. At least part of the molecules go to the nucleus. There, they can modify the splice.
How long is it?
Yeah, we play with that. The main molecule I'll show you is 18 nucleotides long. I mean, in principle, with 17 or so, you can have a unique sequence of the human genome. And so your choice can take that into account. But 15 MERS work, 20 MERS work. And that's also a decision that you make based on the chemistry, because this, I forgot to mention, but the type of modification also affects the stability with which it binds to RNA. Hybrids between this and RNA are more stable than RNA-RNA or RNA-DNA or DNA-DNA.
It doesn't bind to the DNA.
It would bind to single-stranded DNA, but I don't think it'll open up a duplex necessarily. And another feature of this is that there are nucleases that may not cleave the oligo, but they may cleave the target RNA. So specifically, RNase H recognizes RNA-DNA hybrids and cleaves the RNA. This modification prevents recognition by RNase H. So what we want to do is target an RNA, but then we want it to splice, so we don't want it to be cleaved. That would lead to its degradation. So ISIS originally developed molecules like this, but then they would have regions in the middle that lack this modification that makes them a target for RNase H. So they use antisense to knock down gene expression. We don't want to do that. We just want to change the output of a gene rather than reduce its overall expression.
So,
yeah.
And this is not associated with AGO, because another meant to be go to AGO and then close chromatin or.
Yeah, so another pathway, of course, is RNA interference, but that's very sensitive, but that means double-stranded RNA. But here we're forming double-stranded nucleic acid, but these modifications preclude recognition by the RNA interference machine. It's much more sensitive to these types of modifications. You can sometimes put things in the passenger strands, etc., but these are uniformly modified, so the RNAi machinery doesn't recognize them.
You know how long they're going to live?
Yeah, it depends on where, right? So, for instance, in the central nervous system, the half-life in adult mice is 200 days, and in the liver it's 22 days, because, so it depends where. Their nucleases just don't know what to do with it, and they can look at even the decay products. You can find, like, if you start with an 18-mer, you may find a 17-mer, but not anything smaller. And of course, it can be excreted through urine, kidneys, etc. But if you go directly to the CNS, it really hangs out there for a long time. So that's an important feature. All right. So we could either try to guess where these silencers are, or we can search for them systematically, and we chose to do the latter. And so, for example, this is the whole exon 7, which is 54 nucleotides long. And what we did is we made a series of 15 mers that the sequence is such that it would hybridize across the position indicated, and we moved in steps of 5 nucleotides. And then we could introduce this into cells in culture, and then see what's the effect on exon 7 inclusion. And then if we see anything that we like, then we zoom in that region and we make more oligos and look in greater detail. So we looked at lots of oligos, and first, as an example, so we looked within 60 nucleotides upstream of exon 7, and this nomenclature I'll explain. So a green line shows where the oligo hybridizes, and if it's green, it means that it causes more efficient inclusion of exon 7. If it's red, it means that it causes exon 7 skipping. And if it's blue, it didn't do anything. And also the thickness sort of crudely conveys the potency. The thicker line means it's more potent. So we learn a lot from this. This is perhaps not surprising. Any oligos that are very close to the 3' splice site or the branch point sequence interfere with binding of the basal splicing machinery, and therefore the exon is not recognized well.
But wouldn't you expect oligos that are so closely positioned to each other with a close effect?
Yeah.
Quantitative, but not all or none?
Yeah, I know, it's an interesting point. I'll make that when I advance the slide. I come back to this point because, yeah, we actually find that useful. Give me 30 seconds to get to that. Yeah. So I want to say about the blue ones first, that they don't do anything, so that's a negative result. You can only go so far with that. It could mean that they don't hybridize, or it could mean that really there's a neutral region in the RNA that doesn't have an enhancer or a silencer. We're not sure. Let me show you the ones that we synthesize here to make the point that you just asked about. Here's an oligo that causes exon inclusion. If we move it by 1 nucleotide, you get this one. It goes from efficient inclusion to efficient skipping. And our interpretation of that is, this is a known activator called TRAPTA-beta 1, and people had already shown that it recognizes this purine bridge region. And I think what we're doing there is we're essentially a footprint, a functional footprint of this activator. This one probably doesn't affect the binding. We didn't test that, but I'm pretty sure that's what's going on. As soon as you move by 1 nucleotide, you begin to prevent binding of an activator, and you see the overall effect. Now, nobody knows how many proteins are bound to an RNA, but generally these types of proteins, when we've studied them, and others have studied them, they recognize 6 or 7 or 8 nucleotides. So if you have a 15-mer or a 20-mer, it could block the binding of as many as 2 or 3 proteins, and what we're measuring is the net effect. So I think this is a method of footprinting, but crudely. I think you learn quite a bit from these edge effects.
No.
Yeah.
Perfect. So what about the structural effects on the
Yeah, true. That's a good point. I mean, there could be secondary structure, and I alluded to that in earlier slides. And some of these oligos could be acting at that level, and it's harder to study because there's a school of thought that says that, okay, RNA is always structured, period. But another school of thought is that pre-messenger RNA, when it's synthesized, a lot of proteins bind, like H and RNPs that bind single-stranded regions, so they tend to unwind. And you kind of need that unwinding to improve the accessibility, but probably the reality is somewhere in between. So some of these could have effects that have to do with unwinding secondary structure and then changing the accessibility to repressors and activators. Yeah.
And could be possible that any of these changes in the RNA accessibility or whatever change the elongation of the col2, for example, and some of the changes.
Yeah, you'll notice in my talk I don't really address that, but a lot of the splicing has been shown to occur on nascent transcripts, and then the speed of the polymerase, particularly cornblende flavic Argentina, this is why you're asking, and showed very nicely that the speed of the polymerase can influence alternative splicing, and it's possible that these things are acting in part on nascent RNA, and that it can have more complicated effects. I don't doubt that, but my feeling is that that's more fine-tuning of alternative splicing, and we can see some of these effects when we do in vitro splicing in cell-free systems in the absence of transcription. Of course, I mean, quantitatively that's not the same, but the trends of what we see, we can see it that way. And so I think at least in part this is acting directly on the RNA as it's recognized by the splicing machinery. And we also found downstream 60 nucleotides. So, all right, so we're not covering this with uniform density because we got a hit here and then we made a lot of oligonucleotides because these were particularly potent. And so this was probably our lead oligo is one of these. I think I indicate that here. This 18-mer is what's been taken to the clinic. It's called ISSM-NRX. We used to call it ASO, antisense oligonucleotide 1027, because it binds from the position 10 to position 27, so it's an 18-mer. There's a silencer that another group had already described not long before that, called ISSN1, and mutations there are very effective at promoting exon inclusion, and likewise each of these oligos does that. And then we studied that in detail by mutagenesis, biochemistry, mutagenesis, overexpression, knockdown. We identified these repressors that bind to that element. Yeah, go ahead. Two oligos? Right, yeah, we have, and we've never gotten encouraging results with that. We were hoping that there would be synergy, but all we have seen is additivity. But I don't know if that could be the way this particular system works. For instance, the silencer here and the silencer, they may be recognized by the same repressors through some sort of looping, and then you might not expect synergy, but that's always worth trying, and so far it hasn't paid off for us. Okay, all right, let me find a natural break point. Let's see, what else do I want to say about this one? This oligo here blocks the 5' splice site. What we think happens is that any one of these prevents these repressors from binding that recognize a bipartite element there. And then when you do that, then you can measure that U1 binds more efficiently, at least in the test tube, and that's why exon 7 is better recognized. But going back to your secondary structure question, I mean, there could be some sort of secondary structure here, and so we sort of affect the way it's presented to the splicing machinery. We can't be sure. I just want to make a couple of points. The binding site for these repressors is 6 nucleotides. The consensus is like UA, GGG, A, or U. Each of these is not a particularly good match. So the point is that this oligo only binds to one transcript in the cell, namely this one. It's going to prevent these repressors from binding there, but it's not going to affect their ability to bind to any other RNA that they normally regulate. Any other RNA will have, let's say, 1 or 2 of these motifs, but it's not going to have a perfect match to the 18-mer. I mean, it will have a very partial match, and that shouldn't be sufficient for the oligo to recognize to those off targets. In principle, unless you put a huge excess of oligonucleotide, you should preferentially occupy the perfect match, and that's the basis for the specificity of this method. Let's see. This is the chemical structure of that. It's large, so it's 7,000 or more molecular weight, and it's highly charged, which poses some technical issues as far as drug delivery. And okay, this one stuff here, and we'll resume.
We get a 5 o'clock.
where you fractionate, the pellet, and then when they look at the ratio and the effect of the oligos, it can act on that fraction, but also on the cellular effect. Huh?
In the neuroplastic.
Yeah, both. So, I guess probably so, but this is towards the end of the transcript, and these introns are not huge. I think the one upstream is 3 kb, the other one is 700, and so, it's hard to tell there. I mean, I think Release probably didn't investigate in great detail, but, some splicing is post-transcription, and then I think it can influence both, and you sort of build a certain concentration of all of those, so probably it can act on the nascent fraction, but, because some of those experiments are done in glial cells, but in cells, I don't know, more neurons.
לא, אם את מתרגלת, לא. You can't. רצחת וגם ירצת. אבל את יכולה אולי לקחת את זה בגאולה.
אבל זה אסור לביולוגים. זה לא ביולוגי.
נכון.
מקסימום אני יכולה לשלוח אותה.
It's 2 to 4 and twice 45 minutes.
Yeah, but surely then it's synaptic to 4.
In mouse photos, 2:30 to 4.
Why not 2 to 4?
They're crazy.
They start their lecture not at 8, 8:30.
And they hate information.
How come? Because, no, I was sure. Last year I did 2 to 3:30.
Sebastian, you can do whatever you want. You are the teacher, no?
No, but it's already know the building better than I do.
That's the worst thing.
Air longer.
She's in Miami.
Actually, I went to dancing class. I went and I paid like $7.
You need to tell them what is an IND, not necessarily what anyone knows.
Yeah, I plan for that.
Okay.
In the gym. What college?
Okay. Oh no, this is 5 minutes
So
later.
they do.
I can't drink the water.
I don't know, that's really cool because for me too, not only do they help you with your school here. We do have some different things okay.
¿Cómo evitaste crosstalk con
So if you
otras?
do in the app.
You need to check like there is no effect on
target
the
effect. On one hand, it's theoretical, based on randomization, but
That's why you're
15
here.
is quite
that's
No,
more
it's
effective than we use 18. So
I always struggle with the
that was even a good one. That's crazy.
Ah, okay, okay.
Yes, no, but
Pregunta
está válida,
because I'm not eligible for any college. I come from and no, the only difference is that it's not alone.
It's not alone. It's, they have a couple of like merit-based scholarships that are based on, yeah, it's not like FAFSA, which is student aid, scholarship aid. It's not that. This is like
You don't have to pay that.
no.
If I can connect to the network, I can hang out here. Yeah. There's always work to do, so I'm sure you guys are good. Yeah, no, I got some proofs for paper. I dealt with it, but yeah, I want to maybe check with my secretary. Is this I'm all right. I hope you have a strong dose of coffee. So yes, Sebastian asked me about off-target effects, and we chose an 18-mer, that particular 18-mer in part because when we did a blast search, we see that there is no other place in the genome that has a perfect match. If you allow one or two mismatches, then there will be secondary targets. And I think the way to deal with that is that, but we know that if we make an oligo with a few mismatches, it can still change splicing of SMN2. So in principle, there could be off-target effects. But in pharmacology, what you would do is establish a saturating dose of the compound. We're interested in splicing of SMN2, so we can measure at what dose you get maximal splicing, and you don't want to go much above saturation because that's when you begin to occupy sites that don't have a perfect match, and you could have more off-target effects. Now, let's say that some portion of the oligo binds to the wrong RNA. I think you'd have to be pretty unlucky for it to bind in a place that would interfere with splicing or translation or something else. It could happen. then either you figure it out using molecular biology methods, or you look for toxicity. And if the compound is not toxic at the doses that you're going to use pharmacologically, then hopefully you're okay.
And how abundant is the mRNA?
I think we haven't measured that in absolute amounts. It's ubiquitously expressed, and so there's probably some variability. I don't really know if people have figured out how many copies per cell. We detect it readily. Yeah, again, I mean, you establish empirically how much oligo you need to change the splice. One thing I did want to point out is that you can target the exon, you can target the intron. It's better to target the intron because after splicing, the messenger RNA no longer has the binding site for your oligo. If you target the exon, some of them can change splicing, but now, in principle, the oligo could interfere with mRNA export or with translation, and so to some extent we know that those oligos are compatible with translation, but quantitatively they're less potent, and that could be the reason. So definitely that's one reason why we picked an intronic oligonucleotide. All right, so then what you need to do to take this all the way to become a drug. This summarizes a lot of published work, so I won't go into detail. At the beginning, we did experiments with cell-free splicing. Then we did transfection experiments, and this was very new for us. I mean, ISIS already knew quite a bit, but turns out that it's more difficult to transfect oligos in cell culture than it is to deliver them in live animal tissues. That was a surprise to me. It has to do with the number of receptors present. You probably knew that from your work.
I learned it just like you did.
Yeah, so we are traditional biochemists, so we hadn't done any experiments with live animals until we started this work. So we delayed that as long as possible because we were intimidated, and then we were surprised how, had we known how easy that was, we might have done that sooner. So we picked these cells because they're easy to transfect, they're very robust, but these are human embryonic kidney cells. So early on we took a gamble, which is that the gene is ubiquitously expressed, but that doesn't mean that its splicing regulation is identical in all cell types, either quantitatively or even qualitatively. That there could be different activators and repressors, and we might have used an oligo that works beautifully in 293 cells, but it might not do anything in motor neurons. Fortunately for us, that didn't turn out to be the case. And also we felt that we had many oligos that worked, so if we had trouble with one, we could always test another. So then at some point in this process, we decided that we needed to get mice. We used mouse models that I'll tell you about. And then we learned how to correct splicing, showing that it happens at the RNA and protein level. We started playing in peripheral tissues because it was easier, but we sort of knew that sooner or later we're going to have to do that in the spinal cord, and we learned how to do that. And then at that point, we began to measure phenotypes, aspects of motor neuron degeneration, and muscle atrophy, other sort of gross phenotypes, survival, etc. But then there's pharmacological questions about when do you have to give the oligo, early, late, is it the same, does it matter? A required aspect to go to the clinic, in the US at least, the Food and Drug Administration says that you have to have proof of efficacy in 2 animal models. And so other than mice, experiments have already been done in non-human primates, where the only thing you can look at is the pharmacology for the reasons that I explained. The splicing, there's one gene that is duplicated, but it splices perfectly well, so you can't fix what's not broken, but you still have to do some work in primates. This is a very important question that, coming back to what we discussed before, does it only matter that you correct splicing in the central nervous system or the spinal cord in particular? What about muscle? What about other tissues? There is reduced expression of SMN in all cell types, but does it really only matter in motor neurons? This means investigational new drug. So when you want to go to the clinic, you have to apply to the Food and Drug Administration to receive this IND status. That has happened already, and phase 1 clinical trials have started last December. I'll summarize that. But in this case, because it's considered an orphan disease, there's an expedited process, and so this candidate drug has orphan drug status or putative drug orphan status, which is good. Makes it the criteria are a little bit less stringent.
I don't think we covered the concept of
that's kind of a legislative thing in the U.S. When it's a very rare disease, sometimes they don't require the same as if you're dealing with a very, very prevalent disease in order to facilitate. There's no treatment available. They don't want it to take forever because it needs to get something to the clinic. So they have different criteria. Was there a question?
That was my question.
Okay. All right, so let me tell you about animal models. This is a very severe type 1 or type 0 even SMA in mice. So how do we do that? Well, we didn't do it. These other groups have done it. If you make homozygous null in mouse, it's embryonic lethal. So this is a cross between a heterozygote and a transgenic mouse that's transgenic for SMN2. So if you have several copies of the transgenic, you mimic a very mild SMA or up to normal if you have lots of copies. But in low copy, that's what happens here. This mouse has 2 copies of a human transgene and no mouse gene. And so you put it on its side, it cannot rear itself, it's very weak. Actually, this survives less than 5 days. We mostly don't see the progeny because it's very, it's so severe that it's not a particularly useful model. This litter mate is still heterozygote at the mouse locus. So, All right, so we assumed that we needed to target the motor neurons, so these are huge cells. In particular, if we want to change splicing, we have to target the nucleus, and the cell body is in the ventral horn of the spinal cord. Of course, these axons are very long, and they innervate skeletal muscles throughout the body. And the way to target that is through cerebrospinal fluid that surrounds the spinal cord and bathes the brain. So if we can reach cerebrospinal fluid, then the hope is some of the oligo will be taken up by motor neurons in the ventral horns, and that is indeed the case. The way we did it at first is these pumps are implanted surgically, and this is put through a cannula is put through a hole that we drill in the skull to go to the lateral ventricle where CSF is, and that circulates throughout the brain and spinal cord. And the reason is that the mice are so small, but however, this procedure, it's harder to go directly to the spinal cord, the intrathecal space. But even so, this procedure requires an adult mouse because neonate mouse is too small. And so these are early experiments that we did to understand the pharmacology and to study the half-life of the molecule in the central nervous system. So we weren't trying to fix the phenotype. In fact, this particular experiment was done with adult mice that were still heterozygote at the mouse locus. They also had the transgene. So these are very perfectly healthy mice. What we're asking is whether we can correct splicing of the transgene. So when we look by RT-PCR, we use primers that are specific for the transgene, and they don't look at the mouse gene. So what do we do? You put the oligo in this chamber, and it's 100 microliters. The chamber is implanted under the skin on the back, and then through osmotic pressure, the liquid is slowly pushed down this catheter and comes out here, and that, as I said, goes to the ventricle. And so this is you purchase these things. They deliver at 0.5 microliter per hour. So over the course of 7 days, you empty whatever's in the chamber. So what we put in the chamber are oligonucleotide solutions, and such that you deliver between 0 and 150 micrograms per day for 7 days. And then we sacrifice the mice. There's 36. No, not 36. It started 13. There's a bunch of mice here. We sacrifice them on day 8, so this is the day after completion of infusion, and then we measure splicing. That's RT-PCR with a single primer pair in exon 6 and 8, and we do it with few cycles and radioactivity, so we can quantitate, and we do multiple mice, some of which are shown here. And you can see that this is an example from the thoracic section of spinal cord. There's 15% or so exon 7 inclusion, and at the highest dose there's 96 or 97%, so it's a beautiful switch. And protein, we had to, in this case, when we were analyzing heterozygote mice, that's what we had at the time. Eventually we made homozygous null mice with 4 copies of the transgene so that they're still healthy, or very healthy, relatively healthy. And we made an antibody that's specific for the human protein. It doesn't cross-react with the mouse protein if it's present. So this is the low level of SMN that's made from the 15% exon inclusion that you detect in spinal cord. But when we treat even with a subsaturating dose of the oligo, we see a clear increase in SMN by Western blot. Then we asked how long does this effect persist. This was much better than we expected. So we gave this subsaturating dose for 7 days, and then it takes 7 days to deliver that, and then in case there's something left, you can cut the catheter, and so that's it as far as delivery to the CNS. And then we waited 0, 1/2, 1, 2, 4, 6 months. You can see that exon 7 inclusion remains super high. It's beginning to come down a little bit.
How do you explain it?
Yeah, so we consider several explanations, but it turns out to be simple pharmacology. ICES can measure the oligonucleotide concentration in tissues, and they determined that it's in CNS, its half-life is 200 days. So this is what you expect as a result. So in fact, this is an experiment where we're looking after 1 year by immunohistochemistry. And this is an immunohistochemistry with a rabbit antibody that recognizes the phosphorothioate backbone. And it's very hard to make these antibodies, but so you can detect the brown staining. These are motor neurons. And so we do saline versus delivery of that drug. After 1 year, it's clear that the oligo is still present there. And this is immunohistochemistry to the human SMN protein, and you see much greater staining compared to the control. So there should be some protein there from the basal levels of SMN2 expression. This is in cervical spinal cord, 12 months post-infusion. But it turns out the half-life is greater for some reason in adult than neonate mice, so things got a little complicated when we wanted to look at phenotype. First we looked at, this is partly for historic reasons, but we looked at that mild model of SMA. It has 4 copies of the transgene. Now we no longer have the mouse gene. They can still live for 2 years and they're fertile. The phenotype is that it's not well understood, but there's some sort of necrosis of the tail and ears after a few weeks. And so if you look at mice of different ages, this is a control, then you can see that it becomes black at the tip. The SMA mice always have shorter tails, and then, well, eventually they have no tails. And the ears, likewise, look pretty unhealthy. And there's a lot of debate about what this means, and nobody wants to study it in great detail because patients don't have tails. They do have ears, but there are a few patients have been described that have this distal tissue necrosis, and there's 2 papers on this, but apparently it's extremely infrequent. The clinicians tell me that this is maybe not a primary manifestation of SMA, but problems that these kids may have. And these are actually type 1 kids that are now living longer because of very aggressive intervention, and so occasionally this is key. They attribute it to autonomic dysfunction, but also there's vascular thrombosis, some sort of vascular abnormality. It's not well understood. In the mice, though, many different strains and genotypes have been looked at, different severity. Eventually, they get very high penetrance. They get this distal tissue necrosis, and I don't know what to make of it, but that's kind of one of the first phenotypes that we tried to. What we wanted is proof of efficacy of the drug or the putative drug in vivo, and so if there's a phenotype, you try to see if you can fix it. Trouble is, by the time the mice are large enough that we can do the pump, they already have this. They have no tail, so obviously the tail's not going to grow back. And so we started doing so ICV is intracerebroventricular. I showed you infusion, but once we realized that the oligos have long half-life, maybe we don't have to infuse them for a week, so we switched to single injections, and other people have started to do that too. So we do it either in newborn mice or even in intrauterine, we learned this from our neuroscience colleagues, that type of procedure. In embryos, you could do intracerebral ventricular injections. And Ken Sahashi's trained as a neurologist, and he did that in the lab. And we don't do this anymore, but I thought it was kind of amazing that one can get this to work. So you take the pregnant female and you have to exteriorize the uterus, of course, under anesthesia, and then each in the sac, there's all these multiple embryos. You inject 2 microliters into the ventricle just once. And you can use, of course, different concentrations of oligo. And then we saw the same thing. Here we're looking at postnatal day 7 and lumbar spinal cord. You can see the change from mostly exon skipping to mostly exon inclusion. It's a nice dose-response curve. That's what you want to see in pharmacology. You're saturating. And this is what happened to the phenotype. These are normal mice. These are the control saline-treated mice, or we can also use mismatch oligos or scrambled sequence oligos. But the treatment with the Antisense oligo, at least partially rescued or delayed the onset of tail necrosis. This was done with a single 20 microgram injection at embryonic day 15, and now we're looking at mice that are 2 months old. Yeah, correct. There you go. Yeah, they look healthy. So I don't know how this phenotype arises, but this is the first evidence we have that the oligo is able to deal with some phenotypes in vivo, which is a good sign. But then we thought it was time to look at severe SMA. So infusion was out of the question. We used this pretty severe. It's not as severe as the first movie I showed you, because those we could get so few mice that it was sort of hopeless. And this is kind of a nice model that other people have reported. So starting with the same 4 copies of the transgenes, there's 2 on each chromosome. This is that type 3 model. But if you cross it with the heterozygote mice and you go through a series of crosses, ultimately you get mice that don't have the mouse gene and they're hemizygous for the transgene. So one chromosome has 2 copies of SMN2 and the other one has nothing. And then half of the litter are still heterozygote for SMN, so that's these mice. And so you get 50% mice that are like this. They're smaller, so there's a growth phenotype that's well known in the field. Their tails are shorter, and they only live 10 days on average. And this is how they look at postnatal day 7. And here's a bit of a movie. So this is at P7. This mouse is only going to live another couple of days. This is a healthy litter mate. But at this age, I mean, mice are not moving a whole lot. So there is some motor dysfunction, but mainly weakness. It's a little bit hard to tell. So what we do here is 20 microgram intracerebral ventricular injection at postnatal day 1. And this is how it looks in whole spinal cord at postnatal day 7. We've gone from 13-14% to more than 80%. So again, it's very dramatic. This is what's happening at the protein level. We increase full-length protein beautifully. The truncated protein is, like I said, it's unstable, so it's not, you don't even see it. This is what happens to survival, though. So the controls have a 10-day mean survival, and the treated mice, just treated once at P1, have 17 days. At some point, one could become excited by this. It's a very significant, statistically significant effect, but we kind of expected more. So this was a bit of a disappointment. It's been described that transiently there was a collaboration with Genzyme, and they had done this experiment in a less severe mouse model that lives 15 days, but they saw something very similar, and they measure the concentration in tissues over time, and it seemed like the half-life was less than in adults, but also the mice are growing, and so it's hard to fully understand why the effect is limited. Yeah.
How do you compare the levels of HSMN1 to the mice with the 4 copies?
Right. Yeah, so there's been a lot of work trying to figure out how much you need to increase to have an effect. I mean, becomes pretty equivalent to having a whole SMN1 because the transcription is the same, and we're really very efficiently switching. So I can't show you that data here, but I think this becomes comparable to what you get from a single copy of of the mouse gene. I mean, now you're comparing mouse to human, and then the antibody we have, we can't do that comparison, but there are other antibodies that do cross-react.
So you do have 4 HSM2 that you can compare, the one with 4 copies.
Right, yeah. Right.
Which is
Oh, that's what you're asking. Yeah. I don't have that at my fingertips to remember, but I have to kind of look it up. But I mean, in terms of what one needs, so you think whether this may be insufficient because we are not making enough. But I mean, so yeah, there's 2 copies of the transgene. And we go from 15% to 80-something percent. Would that be equivalent to, I mean, I think 1 copy of SMN1, which we haven't put in mice, would be perfectly viable. And it's probably equivalent to the mouse gene, but you know, those are kind of difficult comparisons. So I don't think it's a question of dosing. And by now the standards are high. People have done cDNA transduction with adeno-associated virus, and a somewhat less severe model. They could do much better than this, so we weren't too happy about this. So what we began to ask is whether maybe you just need correction beyond the central nervous system. So this is done by intracerebroventricular injection. So we decided, we already knew that we could deliver the oligo in peripheral tissues, and maybe if we combine the 2, we were thinking that muscle plus motor neurons might be better. So that's what we started doing. We combined, if you look at this curve here, we did 20 micrograms ICV at P1, and then subcutaneously, we also did intraperitoneally with similar results. We gave the dosing, and it's expressed as, in this case, 50 micrograms per gram of tissue, and we give that twice at P1 and P3 to try to build a large dose. Well, it was pretty striking that now the survival, I mean, 50% survival is something like 180 days, and so these are the untreated mice. The ICV alone, I don't have it on the same graph, but it's 17 days. And then if we only gave the peripheral treatment alone, it wasn't as good, but it's still pretty good, more than 100 days. So this you can begin to get excited about, except that how come we're getting a strong result outside the CNS? Well, I'm gonna describe this, but this is in neonate mice. The blood-brain barrier is not fully established, so we began to consider that even when you only give it the periphery, you're reaching both. But this clearly begins to show that maybe you shouldn't restrict yourself to the CNS. Here we show the dose response. Now we only do 2 subcutaneous injections, roughly around P1 and P3. This is each dose, 40 micrograms per gram of tissue, or 80 or 160. The more you give, the better. Now in more recent experiments, we're giving 3 times, and so far the survival looks much flatter. So I think you can completely cure these mice, even though the treatment is done relatively early, but the oligos do have a fairly long half-life. Now the half-life outside the CNS is less. I'm not sure where we should look. In liver, it's 22 days versus 200 days in the CNS. And maybe you just need to correct expression transiently, and then how does that relate to the clinic? I don't know.
Someone did the conditional knockout for it.
Oh yeah, there's been a lot of work on that. Tons of work on it. It's just, the expectation has been in the field that motor neurons is it. And so people have used, recently there's a couple of papers just came out using the chat promoter, the cholinacetyltransferase, that only gives 5-day extension of survival. And then, but still people slowly are beginning to say, well, you need motor neurons and some other neurons. And so they use a prion promoter, which is, that gives very good rescue of the knockout. But if you look at the data in the paper, there's a lot of debate about this. A lot of these drivers have leaky expression. Some of them are the lines that live longer, show some expression in the liver, and typically people look at 2 or 3 tissues, they don't look everywhere. So you think that genetically you should be able to do a very clean experiment, but in practice, some drivers are kind of leaky, so I don't know. But we're very interested in how to address that, so I'll describe our experiments, which are a non-genetic, pharmacological way of looking at it. All right, so we have these 4 doses given only peripherally, and then we take these tissues out at P7. Then we measure exon inclusion, and in each case you see dose response effect. But these are in CNS tissues, brain and spinal cord. Clearly, there's something, so that means some of the oligo is making it to the CNS. It's more effective in the liver, though. And so here, I think this is easier to see here. The 20 microgram intracerebroventricular dose gives a lot of correction in brain and spinal cord taken as a whole, right? So we're just taking a whole spinal cord or a chunk of brain at P7. It doesn't do anything in heart, kidney, liver, muscle. That's a blue line. The subcutaneous high dose does a little bit in brain and spinal cord. It's significant for sure, but it's not as good as ICV only. But in peripheral tissues, it's effective where ICV doesn't do anything. I show you that in survival, this is much better than this. So this is much better than that. To me, that says that, okay, this may be contributing, but clearly you need to do something outside. And it's crude. I mean, we're treating the mouse as having these 2 compartments, and clearly there's leakiness across these compartments, partly because they're neonate mice, but it is what it is. This allows us to look more specifically at motor neurons. I mean, we do immunofluorescence and CHAD positive cells in this part of the spinal cord. These are motor neurons, alpha motor neurons. And then if we look at the CNS injection, ICV injection, and we probe for SMN, there's a ton of protein where the controls, it's below the level of detection. Or in this case, actually, we're showing heterozygote mice, so you can see, I mean, I shouldn't say this, you can see these dots, so these are the gems, so you occasionally find cells in gems. But when we do the subcutaneous treatment, it's no different. There doesn't seem to affect SMN levels in motor neurons, but the CNS delivery has a huge effect. We can count the cells in gems and the number of gems per cell, and that we published that. This clearly increases SMN expression in motor neurons. This doesn't. Phenotype, however, is opposite, the reciprocal of that. But what happens to survival of motor neurons? So we can count motor neurons. This is by CHAP staining. This is where they're located. And there's a reduction. So this is the number of motor neurons in a section in heterozygote mice versus untreated or saline treated mice. This reduction is significant. When we treat with oligo we treat at P1 and P3. Here we're looking at P9. We've preserved the motor neurons. So that's what's shown here. We don't think we're reaching the motor neurons and fixing the defect there directly, but somehow these motor neurons remain healthy. Also, this is a way to look at the neuromuscular junctions, and this is in quadricep muscles at P10. There's a certain morphology that's normal, this synapses, and in SMA mice, it's shown that these are just you lose their integrity over time. They're very abnormal looking. But with the treatment with the oligo, we preserve the integrity of the NMJ, and there's a way to quantitate this. So we also, now the mice are living a long time, we could do these functional tests. These are 3 months old, and so you may be familiar with the rotor rod. This is a task that requires a lot of strength and coordination. And this is a control mouse, a heterozygote. Generally, they do very well. This example is not going to do so well, but statistically, these guys are going to do better. But I'll show you that this is a treated mouse, 3 months of age. It does the task very nicely. And even though we're going to reverse the direction, it manages to hang on, and it has to be very strong to do that. Yeah, now it's in the other direction. Don't worry, it won't another test that I like to show is we take two control heterozygous and two treated mice, treated subcutaneously from different cages, and so they fight because the territorial behavior, this is a P90. And the mice with the short tail are the treated SMA mice, and the others are the carriers. And other than that, well, they move too fast to be able to tell which is which, but they're obviously very strong, and they can defend the territory. So it's pretty remarkable that their theory works. All right, so I want to, these are published experiments and what remains of the time. How do we tell the contribution of the oligos that do cross the blood-brain barrier and make it to the CNS? So I think the field sort of reluctantly accepts that maybe something in the periphery could be important, but nobody's working on that yet. But they can't accept that it's a motor neuron cell autonomous problem potentially. So this is what we came up with. Antisense binds to the SMN2 RNA and blocks the repressor. So we made a sense RNA. We call it a decoy. It's also an 18-mer. It has exactly the same chemistry as the antisense oligo, but it has the same sequence as the target RNA. So if we inject this, it's somewhat in excess. It'll bind to the antisense oligo, and now the repressor can find its site on the RNA. And so the idea is that we take the neonate mice and we do the same as before, subcutaneous injection of the antisense oligo, and then we give the decoy in the CNS. So it should neutralize the effect of that portion of the oligo that crosses the blood-brain barrier. Any questions? All right, so this is the experiment. Then we measure splicing in liver, brain, spinal cord. So what happened here is we've given 3 subcutaneous injections of a fairly high dose, and this is very good for phenotype. But then in this group of mice, we give 30 micrograms of the decoy, and it's given. And first we give 10, and then the next day we give another 20 micrograms. And it's easier to look at the quantitation of the data. In the liver, subcutaneous injection of the therapeutic oligo, and then scaling is a control given in the brain. There's no significant difference here, maybe a little bit. But if we look in brain, the decoy essentially neutralized the effect of the subcutaneous oligo, and the same thing in spinal cord. So that's what we wanted to do. I mean, we're treating the mouse as having 2 compartments. When we give this, there's a bit of leakiness going across the blood-brain barrier, but we can take care of that by injecting the decoy oligo in the CNS. And the question is, what happens to the phenotype? Any predictions? All right, so this is what we find. So this is with the decoy. You can see there already the experiment's been going on for 150 days. It's a little bit less than that, but this difference is not statistically significant, or at least not yet.
But what about the peripheral phenotype, the Golgi?
So which, you mean the distal tissue? Well, so yeah, I didn't mention that, but what has happened to our mice is that we turned them from type 1 into type 3, but very mild type 3. So eventually they get necrosis. I mean, you saw the one in the rotor rod didn't have much of the tail or the mice fighting, but the onset of that is, and maybe this gets at your question, it's less severe than the type 3. So the type 3 has 4 copies of SMN2. They develop necrosis after 3 weeks. Our mice develop necrosis later than that. So, yeah, this is pretty remarkable. So I think this kind of is saying that as a first approximation, it says that the oligo doesn't need to act directly on the CNS, but there could be some specific groups of cells that don't represent the vast proportion of where we can measure splicing. Because here, what we've done here is we've taken a chunk of brain, chunk of spinal cord, but there could be some particular cells somewhere that are very important. If so, we need to know what they are. Yeah.
How do you know when you inject the decoy that you
I mean, we can't look at motor neurons. Motor neurons, I showed you before, we cannot even detect. I think oligo is getting through, but I don't really think it's ending up at motor neurons because we don't see an increase in the protein, and we haven't done like laser capture microdissection to look at RNA and so on. But this is what tells us that it should be enough. I mean, it's back to the control levels.
And the behavioral tests on these mice are fine also, not just the...
Yeah, we haven't yet done broader rod and so on. Right now we're following survival and then observations that yeah, I think it's fine. So we do need to look at that in detail.
You know why they died? I mean,
no, cause of death. I mean, people talk it's likely some sort of cardiovascular failure, but it's a little bit hard. No, it's sudden death, so you don't see it happening. It's hard to know. Yeah.
Can't the decoy bind to the actual protein just like
Yeah, that's a good question. The protein binds RNA, but actually it's shown the structure that years ago we studied that protein and we showed that it can bind telomeric single-stranded DNA because the telomeres have single-stranded overhangs and they have that same sequence, DPA, GGG, and that protein loves to bind to that. So can it bind to modified oligo? We didn't really determine that, but it's possible, and that may be why this is perhaps not as good as that. But I think the way the experiment came out is fine. I mean, if this had looked like that, we might have misinterpreted the reason for it. But I don't think it's an issue. I mean, HRMP1 and A2 are very abundant, so it's possible that they bind to the decoy and that causes off-target effects. But the main result here is that these mice are doing quite well. So I don't think it affects our interpretation.
and also like
Yeah.
going way back
Yeah.
the mechanism, but shouldn't you see a little bit of info inclusion of the infant before exon 7?
Yeah. You're possibly right, but we didn't specifically probe for that. I mean, that's a 3 kV intron, so we wouldn't pick it up by the PCR that we did, but you can measure that specifically to put a primer in exon 6 and a primer in intron 6 and see if it's accumulating. Frankly, we didn't check, I mean, because this is more, we're mostly concerned with whether we're increasing the product, and we are, but you're correct about that.
But in all the PCR results that you show,
Yeah.
so
Yeah, we don't see that. I mean, if I were to show you the whole gel, for instance, for the 293 experiments, you do see some bands up there, but I mean, I don't think you can. It's not quantitative because if you have a 3 kV intron, it's a much larger product. It doesn't amplify as well. So I mean, right? That's not what we're looking for, but you can see some evidence of that in some of our published earlier work. I mean, also there are experiments where we start seeing skipping of exon 5 and exon 7, but you wouldn't see that in this primer design.
So basically on the protein level,
Yeah.
how much activity of the protein can you restore?
Well, I mean, I've shown you some gels. Yeah, like here, right? I mean, well, we want it to increase, right? Ideally, we want to increase a lot, but even when you look at the normal human population, it's variable because people have different copy numbers of the normal gene and so on. So clearly, we should increase to whatever extent we can, and that's happening. Right? I don't know if we're, and also what we do is when we do dose response, we know that putting more oligo is not going to help. But getting more complicated than that, I mean, if you want to maintain high levels, maybe you have to keep giving oligo at some frequency. We haven't done those studies in great detail, and I think that's more for the clinic because mice are different from people in pharmacology. So those are very important questions that you need to get at eventually. How frequently do you dose? Where do you dose? And of course, we can sacrifice mice and measure, but in people you can't do that, and so you have to have some indirect measure of phenotype, or even we don't know where to look for that. If it's going to be in the spinal cord, if the peripheral expression observations are relevant to the clinic, then maybe you can measure it in blood. But people have been looking at patients like type 1, type 2, type 3, type 4, and looking has evolved to be efficiently included in the case of SMN1. It doesn't mean that that has no repressors. We still have repressors, but it's the overall effect. Let's say when it evolved, it accumulated mutations here and there, so that at some point it becomes actually efficiently included. That's all we need. There's still repressors there. If we target SMN1, we're not, because mice don't, the people, for instance, that you would treat don't have SMN1, but you cannot make exon inclusion more efficient than if it's already 100%. So I think you would displace that repressor, but it doesn't matter. It's already efficiently spliced. All right, so I'm going to try to give you some explanation for how these things could be acting in the periphery. Keep in mind that we haven't proven that this is important, but it's an observation we made. We thought that since these mice are small, that there's a growth phenotype. We measured growth hormone in the circulation. We didn't see any problems, but we looked at that axis, and so insulin-like growth factor 1 can influence growth. It also has neurotrophic effects, so we measured it. And this wasn't known before, but we found that in the SMA mice, the levels of circulating IGF-1, which is made in the liver, are greatly reduced compared to the heterozygotes. And moreover, when we did the subcutaneous treatment, we restored the normal levels.
Do you have an idea of the mechanism, how this is going on?
A little bit, but no, the answer is no. So, then we measured mRNA, so now we're looking at the mouse genes, right? So the thing is, IGF-1 in the circulation is in a ternary complex, and these binding proteins called BP3 and acid Laval subunit have been shown to be very important to stabilize the growth factor. And if we look at the IGF-1 levels, we looked at postnatal day 1 and 5 and compare heterozygote to SMA mice. It doesn't change. At P5 it goes up, but it's the same in hets and sick mice. But the acid Laval subunit is reduced at P1 and at P5, and BP3 is not changed. So we don't know the mechanism of that. This could be splicing indirectly because of the reduction in SMN levels. The mouse gene only has one intron, and we didn't see accumulation of the pre-mRNA, but we have to look with more sensitive methods. I mean, there is less mRNA, that's what we know. It could be transcription, it could be RNA turnover, maybe splicing. But still, I mean, how we go from reduced SMN to this, I have no idea. But this kind of explains why there's less circulating IGF-1 protein, even though this message does.
Can I make a suggestion?
Yeah.
The SMA kills cholinergic neurons, so you will have reduced levels of acetylcholine.
Yeah. Okay.
And that would elevate inflammation.
Right.
And that could affect the IGF-associated protein.
Do you think through inflammation you expect it?
I think that the effect of inflammation now.
Yeah.
Profound.
Okay. So do you have a suggestion for how to test that? I mean, we could cause, or we look at another inflammatory condition.
I'll take a look after you talk.
Perfect. Okay. I'm hoping to leave with information like that. So anyway, we hint about mechanism, but not at the molecular level. But what we see is that when we treat the oligo, IGF, acid Laval subunit mRNA goes back to normal levels. Now, I can imagine how this could be in the mice and not in people, because, well, first of all, we don't know how it's happening, but whatever splicing or RNA turnover or transcription, is it conserved? We asked clinicians to now measure IGF-1 in children with SMA, and they're doing it, but we don't have information yet.
In blood, or is it
Yeah.
in the blood?
Yeah, this is circulating IGF-1. Now, this factor, this would be expected to go to CNS, and it has neurotropic effects. At least on paper, this looks plausible. Technically, it becomes very hard to prove that because, you imagine, well, let's inject IGF-1 or do it genetically, but there's some other clinical trials have been done for Lou Gehrig's disease, and they have failed, because of the instability of IGF-1, and it has to be formulated in a special way, so I'm not sure what we are able to do. Also, I mean, this is just a correlation. At a minimum, I would say that IGF-1 could be a useful biomarker if it's the case that you see that in patients. And then when you treat the oligo, if the levels go up like they do in the mouse, Anyway, so this may not be necessary, may not be sufficient, there could be lots of things. It's just a way to think about how you go from the periphery to the central nervous system. So just to summarize that, this putative drug, when we deliver it to the CNS, it corrects SMN2 splicing. It has a therapeutic benefit, but it's modest, and we've shown that in mouse, in mice with severe or mild. I showed you that we can correct the tail necrosis phenotype, and we can slightly improve survival. But when we deliver it systemically, there's a much greater therapeutic benefit. I showed it to you for that. It's actually true for the necrosis phenotype as well. We used to think, and we were puzzled, how do you fix that distal tissue necrosis through the CNS? But actually, we do even better systemically. We haven't published that yet. This correction, I think, is transient because if we look at least in the liver over time, the effect sort of diminishes, but by then there's already been a major impact, at least in survival and also in the motor tests. Somehow this, I think, is an indirect effect of SMN levels in some peripheral tissues that then impact motor neuron health. To me, this data says that SMA may not be a cell-autonomous defect of motor neurons. Then I show you this about IGF-1, but I don't know if it plays a direct role in pathogenesis, nor do we know how relevant some of these findings are to the clinic. As far as where we are in the clinic, this will seem a little paradoxical because, I'll get to it in a moment. So what you need to do is to follow through all these steps, and one of those is to use cynomolgus monkeys to show that the drug, how do you deliver it, and how does it distribute it. And the whole thinking was that we need to correct it in the CNS. This is a process that took a couple of years. We didn't have the results I showed you, and it still remains to be determined if it's going to be like that in monkeys or in patients as it is in the mice. So at the moment, that's been more or less ignored. So IND approval was obtained, and the phase 1 trial started in December. And it's being given as a single intrathecal injection. That means by lumbar puncture, L1 and L2, pretty much like you do a spinal tap to test for meningitis or something. And so this is open label, which means there's no placebo controls, or because it's an invasive procedure, and so you don't, it's not ethical to just do a spinal. So in any case, phase 1 refers to a trial just to test where the drug is and that you can safely deliver it and that there are no adverse effects, to establish what kind of dose might be useful or tolerated. You don't look at efficacy, that comes later. So this is what they decided to do, is to take patients that are ages 2 to 14, which pretty much excludes type 1 patients, very severe. Also patients that are not on ventilatory support. So it's mostly type 2 and type 3 SMA patients. And each cohort has 6 patients, and they inject either 1, then 3, 6, 9, but in different groups of 6. Later there will be multiple dosing. And right now they're around here. So far there are no adverse effects that I'm aware of. There's these 4 centers, but it's not clear that they're going to participate because even though you have FDA permission, then each institution has to give approval, and some places are more bureaucratic than others. So they were first. This is at Columbia. They were second. They have started, and probably it'll be finished before they get their approval. And then also this big biotech company, Biogen, became interested in this, so they are funding all the clinical trials as long as things go phase 1, phase 2, phase 3.
So you have to apply for a new IND for this one injection?
Yeah. Right. So, but I've been insisting, that it would be a lot easier, and you could also argue that, okay, maybe that's not going to be like in mice, but there is defective gene expression in the periphery. If you don't do that, it's possible that even if this works, then you're delaying problems. Later on, something else is going to show up like that, necrosis or something worse, cardiac failure. So the thinking is to start adding, continue with this if it's going well, and then do plus or minus peripheral injection. But the that could be later this year. This phase 1 trial will be completed this year in a few months. Okay, I think that my time is up because I have another story that another time. Yeah.
Do you know if this now if the two genes that you follow
Yeah.
have the same pattern of expression?
Yeah, pretty much. Yeah, there's a lot of homology to promoters, so all the data that I've seen, they go in parallel. And we haven't done a lot of that, but when we use 293 cells, for example, they have both genes. So we have an RT-PCR trick with a restriction enzyme digestion that we can see both products separately. So we could see also that our oligos, the SMN1, when I say it's constitutively expressed, it's not 100%, it's like 98%. And the oligo could fix that, and pretty much went together. And we pretty much saw the same levels of expression in the cells that we photographed.
Yeah, specifically in neurons, because this is the, if they're not,
Yeah.
then all this is not.
Well, but I mean, so people are doing experiments like iPS cells and then deriving